What is capsaicin? You've probably felt that burning sensation inside of your mouth when you have a chili pepper, any kind of spicy thing. That's capsaicin. You've probably also heard of the Scoville scale, the scale of how spicy a chili can be from least spiciest to most spiciest. Most spiciest on the Scoville scale is pure capsaicin. It's a very interesting molecule, but how on earth can plants produce such an interesting thing that can do so much odd stuff inside of their bodies, which I'll go through in a separate videos. And in plants, capsaicin can be produced from amino acids. Now, amino acids are things that can be digested when protein's eaten inside of humans, but hopefully if you're at the age where you can watch YouTube and actually click on my videos, you may figure out that plants don't have mouths and they cannot eat. So how on earth do proteins get produced inside of plants? So in this video, which is going to be a very, very long one, by the way, if you may not have already gathered, I'm going to see how on earth both phenylalanine and valine, the proteins and amino acids that are used to produce capsaicin, how they are made, and then how they are made to produce capsaicin. And the first step inside of the reaction to produce phenylalanine is phosphoenol pyruvate plus erythose 4-phosphate as well as water produces DHAP plus PO4, an inorganic phosphate group, and this reaction is catalyzed by the enzyme DHAP synthase. And because both phosphoenol pyruvate and erythose 4-phosphate have a phosphate group, that's why the inorganic phosphate ends up being excreted as waste or whatever. But how on earth are those two molecules created, phosphoenol pyruvate and erythose 4-phosphate? Now, these two molecules, phosphoenol pyruvate, you may recognize from when I've talked about glycolysis in past videos, and glycolysis is something that happens inside of plants as well. Plants produce glucose during the process of photosynthesis. They'll then break that glucose down to produce energy, and you get phosphoenol pyruvate along with that process, which can then be used in a separate process to be combined with erythose 4-phosphate to get the phenylalanine in a few future steps. And erythose 4-phosphate is an intermediate inside of the Calvin cycle, which is what happens inside of plants. It's the non-light dependent reaction stage of photosynthesis. It's a bit where I'm not really so sure what exactly happens. I'm not really sure or educated enough on the Calvin cycle. I know it's just produced during then. Plants do produce it. It's a very well-known process by people that study plants a lot, I guess. And the second step inside of the production of phenylalanine inside of plants is that DHAP will end up being converted into 3-dehydroquinate, and that is catalyzed by the enzyme DHQ synthase. And then the third step is you take that DHQ and it will end up turning into 3-dehydroshikimic acid. And this reaction is catalyzed by the enzyme 3-DHQ dehydratase. Now, a lot of the step 2, step 3 reactions haven't had many coenzymes. That's why I've been able to get through them so quickly. But a lot of the reactions in here can get very, very complicated, like the next one. And the next one is step 4. You take that 3-dehydroshikimic acid, you add it to an NADP plus molecule. That will end up turning into NADPH plus shikimic acid. This reaction is catalyzed by the enzyme shikimate dehydrogenase, I'm pretty sure. And the NADPH and NADP plus, the NADPH turning to NADP plus essentially means this reaction ends up consuming energy. And that is why processes like glycolysis and the Krebs cycle exist inside of nature, so that your body can end up using the energy to actually do other things, to produce things like capsaicin, which in plants is needed as a defense mechanism for animals to not eat them as much, or when they do eat them, it will end up being a deterrent for them to eat them in the future because it's such a spicy thing. Humans seem to like that kind of stuff. We've evolved to not really care about capsaicin. We know scientifically that it's not a toxic thing. It doesn't actually do any cell damage or anything like that. Even though it may seem like quite an inflammatory substance, it ends up not being a thing that inflames your cells or tissues at all. And so humans end up living on the edge and decide to eat capsaicin because they like that spicy feeling, I guess. And the fifth step inside of the production of phenylalanine is shikimate plus ATP converts into ADP plus shikimate 3-phosphate. This reaction is catalyzed by the enzyme shikimate kinase. Hopefully, if you've been watching my channel for a while now, hopefully you may understand things about kinases, ATPs, ADPs, but if you're new, I would highly recommend watching my other videos that I have made on those sort of things, which, while may seem quite basic and boring to me right now, the sort of ATP, ADP, shikimate kinase, etc., that may seem like quite an interesting thing for you. So I highly recommend checking those videos out for you to be able to understand it a little bit more. And the sixth step inside of the production of phenylalanine is you take that shikimate 3-phosphate, you add it to another phosphoenol pyruvate molecule, and that will end up turning into EPSP plus a PO4 inorganic phosphate molecule. Not sure what EPSP stands for, I'll put it up on screen, but this reaction is catalyzed by the enzyme EPSP synthase. And the seventh step inside of the pathway is you take that EPSP, you convert it into chorismate plus an inorganic phosphate, and this reaction is catalyzed by the enzyme chorismate synthase. What's quite funny is that this pathway from phosphoenolpyruvate and erythose 4-phosphate all the way down to phenylalanine ends up taking quite a long time. It's almost longer than the actual sort of production of capsaicin itself, which is, yeah, that's another pretty interesting thing. And the eighth step is chorismate will end up turning to prephenate using the enzyme chorismate mutase. And the ninth step is prephenate will end up turning into phenylpyruvate using the enzyme prephenate dehydrogenase. We've got the phenyl part now we just need the alanine part. We're going to be almost there. There's only one more reaction step to go. I'm a little bit worried during the scripting of this video that I may have missed out a few coenzymes, because there wasn't really sort of a set pathway that I could find for the production of phenylalanine, as I haven't been able to find for a lot of the research I've done for some of my videos. So if you are sort of more advanced in science than I am, please do sort of tell me if I've missed anything out, because I would like to know, because some of these reactions do seem a bit boring in a way. It's just one thing turns into another using an enzyme. There aren't any coenzymes or anything else involved, which would be nice to know if there actually is, and it actually is more complicated than it sort of seems, like one molecule to one molecule using an enzyme. I would be very interested to know if that is the case. And I've just had this knowledge sort of disbarred from me, and I need to go through many more years of university and education to actually be able to sort of learn and understand what that kind of stuff, those coenzymes, those really peculiar molecules, proteins, etc., actually are. And the 10th step is you take that phenylpyruvate, you add it to a glutamate molecule, and that will end up turning into phenylalanine plus alpha keto glutamic acid. The reaction is catalyzed by the enzyme PP amino transferase. Alpha ketoglutamate acid will probably then end up going to the Krebs cycle. I do assume that the Krebs cycle does happen inside of plant cells as well. While they do use photosynthesis to get the majority of their glucose and energy, that glucose will still end up being sort of digested, not really digested, but metabolized, go into acetyl-CoA. And I assume that plants and stuff do have the sort of necessary facilities to be able to facilitate the cycling of the Krebs cycle, I guess. And I assume that alpha keto glutamic acid or whatever it was will end up then going into the Krebs cycle, just as a lot of other intermediates do, like that sort of phosphoenol pyruvate will go into glycolysis, etc., and all that sort of stuff. But now we do finally have phenylalanine, and that took almost 10 steps. I promise none of the other sort of metabolic processes. I think the production of valine may end up only taking like 3 to 4 steps, and then after that, phenylalanine and valine, when they are converted into the molecules that then combine, that also only takes like 4 to 5 steps as well. So it shouldn't be too long from this point onwards. But yeah, hopefully you do enjoy this so far. And so now I will go on to the sort of production of valine, and the first step inside of this metabolic pathway is you take 2 pyruvate molecules, and this will end up going and turning into 2-acetolactate plus CO2, and this reaction is catalyzed by the enzyme acetolactate synthase. And the second step is you take that 2-acetolactate and you turn it into 2,3-isovalerate. There may be an enzyme that catalyzes this reaction, but I probably couldn't find it during my research because, as I've said, I did struggle with finding a set pathway to be able to sort of find this. I had to use a lot of different sources to get different parts of the pathway in, which I hope now I'm making this video as I speak. There is now going to be like a set guide to how capsaicin is actually produced inside of plants that is as detailed as I can possibly sort of make it. And the fourth and final step, you'll notice that this one is a lot shorter than the previous one we did, is you take that 2,3-hydroxy isovaleric acid, I think it was, and this will end up turning to valine using the enzyme dehydroxy acid dehydratase. A lot of Ds, a lot of hydroxys, a lot of acids, a lot of hydratases sort of things. Loads of sort of complicated names and words for molecules in chemistry and biochemistry, which is cool. But hopefully if you're watching this video and this is the first time that you've been introduced to a lot of the sort of biochemical side of the world and the world that we live in, hopefully I've not confused you too much and tried to keep it as simple as possible while sort of being as detailed as I possibly can. Now you finally have valine and phenylalanine, and these two molecules can go through two separate pathways and then be used to produce capsaicin right at the very end. I'll go through the production of phenylalanine into its sort of molecule right before it turns into capsaicin, and then I'll end up going through valine, and then it's time for the final reaction step, which can produce capsaicin. And the first step inside of the pathway from phenylalanine to the sort of molecule before capsaicin is phenylalanine ends up turning to cinnamic acid, and this reaction is catalyzed by the enzyme phenylalanine ammonia lyase. This reaction ends up getting rid of the nitrogen group from sort of phenylalanine. As I've said before, amino acids contain nitrogen groups. That's why they are amino acids, because in organic chemistry, an amine group contains a nitrogen. And the reason that it's ammonia lyase is because when that nitrogen group gets taken off the phenylalanine, the enzyme probably sort of carries it in all of its sort of protein-shaped wiggles and jiggles or whatever, and that ammonia will end up being gone off, and it will end up being used to sort of do a few different things. I mean, I know in humans, when a protein is sort of deaminated, it ends up being used to produce urea, which gets excreted by our sort of urinary system, but I'm not actually sure what happens to that sort of NH3 group once it ends up being deaminated inside of plants. And the second step is you take that cinnamic acid, which is commonly found inside of cinnamon, hence the sort of similarities inside of names. You add it to molecular oxygen, NADPH plus an H plus ion, and this will end up turning into coumaric acid plus NADP plus H2O. And the enzyme that catalyzes this reaction is cinnamic acid Cl hydroxylase. And the third step inside of this metabolic pathway is that coumaric acid ends up also getting added to molecular oxygen and NADPH, and that will end up turning into caffeic acid plus NADP and H2O. This reaction is catalyzed by the enzyme, I think it's coumaric methyl transferase into caffeic acid, which is commonly found inside of caffeine, coffee beans, all that sort of stuff. Hence why you sort of see how plants utilize amino acids and proteins to sort of make the molecules that make different sort of plants and plant-based foods actually sort of unique. You've seen cinnamic acid, and now you've also seen caffeic acid. You see where these sort of plant metabolisms and stuff end up stopping in like a load of other things, because certain plants may have mutated and may not have the enzyme to sort of carry on producing the molecules that it needs to produce capsaicin, and so they've evolved to end up just being coffee beans or whatever. It's a whole very interesting thing. And the fourth step is you take that caffeic acid, you add it to an SAM molecule. Can't remember what that stands for, but I will put it up on the screen or whatever. And that will end up turning into ferulic acid plus an SA molecule. No M this time. And this reaction is catalyzed by the enzyme caffeate methyltransferase. And the fifth step is kind of two steps in one in a way. The first sort of part of the fifth step is ferulic acid plus oxygen. It goes through a process known as oxidative cleavage. That does something, I guess, cleaves off some part of the molecule. That will turn it into vanillin. You may sort of see ideas as to where this is sort of going. And that will end up turning into vanillylamine using the enzyme aminotransferase. I don't know why they couldn't just sort of, in the sort of evolution and metabolism of this process, they couldn't just keep the nitrogen on for the entire time. Maybe they do actually do that, and I've just accidentally somehow managed to do my research completely wrong, and this is actually a completely wrong metabolic process. I really hope that's not the case. I really hope I have got everything right in this sort of process. But I guess nitrogens are very hard to work with in a sort of chemical sense. They're not the most reactive things. They're going to be very hard to move, especially if you need that amine group, that nitrogen group, to actually be moved to a different position on the molecule. That's going to be very different, so it might be better off just taking it off entirely and then adding it back on at the end. I assume plants are quite smart, and that's just how they sort of logically thought and figured that out. But now you've got vanillamine. That's the final molecule. That's the molecule that will be combined with another molecule that valine produces to produce capsaicin. If you look at a molecule of capsaicin, you can see how like the little head bit of capsaicin looks a bit similar to vanillamine, and that will end up converting with the molecule that valine produces to make capsaicin. And the first step inside of valine end up turning into the molecule before capsaicin is valine turns into alpha keto isovalerate using the enzyme BCAT. The AT part of BCAT, 99.999% sure that stands for amino sort of transferase or whatever. That's going to get rid of the sort of amino group from valine to actually turn into alpha keto isovalerate. Alpha keto acids are acids that are sort of, they're carboxylic acids, but they've now had the amine group removed and they have a ketone group sort of opposite the sort of carboxylic acid sort of double oxygen bond. There's things like beta keto acids, gamma keto acids, depending on where the sort of oxygen double bond is. But that is essentially how alpha keto, beta keto acids do actually work. Now, the second step inside of this pathway is alpha keto isovalerate ends up turning into isobutyric acid using the enzyme alpha keto dehydrogenase. I really struggled to find a lot on this reaction step as well. So when I was sort of looking for things on Google, Wikipedia, loads of other sort of science, I think there's things called like ResearchGate, ScienceNet.com or whatever, I couldn't find anything on this reaction step. I had to refer to good old ChatGPT to actually help me find the coenzymes that may be involved in this reaction. All it gave me as an answer was various coenzymes. Didn't specify what they are, but I assume various doesn't just mean one or two and may just sort of mean quite a lot, like 4, 5, potentially double digits, to actually sort of make this reaction work and to end up meaning that capsaicin can be produced inside of plants in the end. Now, in the third step of this metabolic pathway, a coenzyme A molecule is added onto the sort of isobutyric acid molecule, and this will end up turning into isobutyrate coenzyme A. And at this point, loads and loads of methyl groups are added onto the isobutyrate CoA molecule, one at a time, I assume, in each individual chemical reaction, enzymatic reaction, and that ends up turning into a complicated molecule which includes numbers, which is never a good sign when you're sort of working with organic chemicals, which is 8-methyl 6-nonoic acid, and yeah, loads of numbers, loads of suffixes and prefixes. It's not got a nickname for it, but that 8-methyl 6-nonoic acid will end up being added to vanilamine to make capsaicin using the enzyme capsaicin synthase. You have capsaicin. The chili peppers can now withstand a few sort of evolutionary hits from predators. Not humans, of course, because humans seem to have loved spice over the years, even though many other animals that live in the wild will not fancy chili peppers just as much as we do. But that's it. That is how capsaicin is metabolized in plants from, I think there was 2 pyruvate molecules to make valine. It was a phosphoenol pyruvate molecule and erythose 4-phosphate molecule to make phenylalanine. Those 4 molecules end up being metabolized into capsaicin. How cool is that? And it's taken a while for me to get here. I think it's 30 minutes at least on the sort of top of the screen that I'm recording on, but that may end up being cut down to around 25 or so, considering all the breaks that I've had to take, because I'm not used to recording for this long of a time whatsoever, and my throat is very, very sore right now. So with that, I hope you enjoyed, and I also recommend subscribing because I love this community of young tribal people who are willing to improve themselves so much more than they already have, and good luck.